Vivek sir, I am making you as a host of this particular session, and thereafter I will leave.
Okay, sir. Okay.
Okay. You're live.
Okay. Shall I start?
Yes.
Okay. Thank you, sir. So, good morning, all of you. I, Dr. Vivek Srivastav, thank you all for joining us today for the webinar on clinical drug development and role of clinical pharmacology. Honorable Dr. M.N. Patel, sir, Provost, Parul University. Respected Dr. Abhay Dharamsi, Dean, Faculty of Pharmacy and Principal, Parul Institute of Pharmacy. Dr. G. Chakravarti, Principal, Parul Institute of Pharmacy and Research. Dr. Lalit Lata Jha, Madam, Principal, School of Pharmacy. Please join me in welcoming our distinguished speaker, Dr. Satyendra Suryavanshi, Director, Clinical Pharmacology and Pharmacometrics at Bristol Myers Squibb, Princeton, New Jersey, USA. So before I hand over proceedings to Dr. Satyendra Suryavanshi, I would take a moment to introduce all attendees to Dr. Satyendra Suryavanshi. Dr. Satyendra Suryavanshi earned his B Pharm and M Pharm from Dr. Hari Singh Gaur University Sagar and a PhD from Central Drug Research Institute, that is CDRI, in affiliation with Jawaharlal Nehru University, New Delhi in 2008. And he completed his postdoctoral fellowship in clinical pharmacology and pharmacometrics at University of Tennessee Health Sciences Center, Memphis. For the past 10 years, he has worked in BMS oncology, cardiovascular, and metabolic clinical drug development. He has contributed to the development of biologics and small molecule drugs as a life-saving treatment for individuals with cancer, cardiovascular risk, and diabetes. Recently, he has been awarded with highest R&D award of BMS for developing an anti-cancer drug. And I would like to tell all our attendees that we will be having a question answer session at the end of this webinar. And if don't worry, if you miss out something also, a recorded version of this webinar will be available at Parul University's Facebook page, that is PU Webinar. So with this, I hand over the proceedings to Dr. Satyendra Suryavanshi. Sir, welcome, sir.
Thank you, Vivek, for nice introduction. And I am very happy to present some of the expertise that I have gained over the period during my PhD, postdoc, and during my industry tenure. And it's always nice to bring back the learning to the students early in their career and see some of these learning what we have gone through long career can bring them and introduce to some kind of very interesting field which are not very much known back then when we were doing our undergrads program. So I am very happy to present and share some of these learning with new generation, new pharmaceutical scientists. So my talk, as Vivek introduced, it will be providing an overview of clinical drug development in the industry setting, as well as highlight the importance of clinical pharmacology in clinical drug development and what it means by developing a clinical pharmacology package for any new drug development. So in my presentation, I will touch upon overview of clinical drug development. I will define the clinical pharmacology function. I will also speak on some of the key differences between different drug candidates. Sometimes these can be a small molecule or the large molecule, and how the clinical pharmacology strategies differ between these two different sources of drugs. We'll also go into what is needed for a successful registration from clinical pharmacology function. What kind of studies we need to conduct, and what really it comprises, and what is the end product when a drug finally get registered, what is the end product in the label, how the clinical pharmacology findings are presented in the label. And I will also present some case studies, cabozantinib, which is already an approved drug. I will also bring some of the real application of these clinical pharmacology when you go beyond the registration. So in post-approval setting, how these clinical pharmacology characterization can help extending the label, as well as extending the label to the new indication or the new dose regimen, or let's say new special population such as pediatric patient population. So what you're seeing in this slide is an overview, which I'm sure basic curricula in pharmaceutical sciences covers this. But in the industry setting, this is basically this whole continuum of drug development is pretty much divided between the preclinical and the clinical space. So research and discovery, most of the time, there are dedicated functions which are screening many different candidates, sometimes thousands, tens thousands of candidates screening to find a good hit in terms of, based on their preclinical model, based on their some hypothesis. These are well defined. And many of my part in India, like when I was in Central Drug Research Institute, Central Drug Research Institute was pretty much in this this space where we were trying to see various sources of drug from the chemical point of view, biological, or the marine or the herbal sources. So it has a vast, basically sources of candidate optimization within different functions. So same here in industry, you will have that research discovery function. Once you have a good hit or good lead molecule, you will try to progress that in the non-clinical or the preclinical space. And pretty much from there, from that point of time, most of the information what you are generating in non-clinical space will be very important for the clinical drug development. And when we say about, there is a new term called translation medicine. Basically, the idea is the learning what you have in the preclinical space, how can you extrapolate to the clinical and inform your clinical drug development, so to speak. So in the preclinical, non-clinical, again, in this, it's a dedicated function which will try to evaluate non-clinical safety, understand different in vitro, in vivo target, profiles. And again, depends on what is your drug candidate is it a small molecule or large molecule? These in vitro, in vivo package will vary a lot. In case of a small molecule, there is a whole host of the in vitro studies one has to conduct depending on the liability with the given asset. For example, if let's say you have a molecule which is low soluble, low permeable, those kind of characterization has to happen early on. Similarly, a small molecule can do different drug-drug interaction. There is a, let's say, PGP substrate inhibitor or CYP inhibitor or inducer. All those characterization at the in vitro setting has to be completed before the molecule transferred to the, based on, again, the safety profile and all, it can transition to the clinical development. And most of the time before we do the phase 1 study, we will put together an IND package, which is primarily, the content of that is primarily the non-clinical characterization, as well as the proposed phase 1 design and what is the first in human dose recommendation. First in human dose recommendation is another topic which can go in very detail, and very different approaches can be taken for a small molecule and large molecule, which I will discuss later on. But again, all of these different stages of drug development is well supported by regulatory guidance. I'm sure you may be aware there are different regulatory agencies around the world, primarily US FDA and the EMA from the European agencies. they are the primarily has a very robust guidance around all of these different stages of drug development. So I will encourage, highly recommend to go through those guidances to really understand how the non-clinical study should be conducted, how the in vitro characterization, in vivo characterization should be done, as well as how what are the different strategies for first in human dose projection. So these actually, that's the most, I would say from the academic point of view, it's always good to look from what is the highest standard one has to establish in terms of making a new drug to register, and then take it from there and bring those learning to your academic setting when you're doing some experiments. So that's always good to know the current best practices in conducting these experiments. So once we start doing a phase 1 study, again, the primary objective for any of these phase 1s are primarily is the safety. So first we establish the dose safety relationship. We want to establish the maximum tolerated dose one can achieve. In case of different therapeutic area goes with different patient populations or healthy population. So in oncology, for example, we will typically conduct the phase 1 study in the patient population. So it's very different than what you will do for example, rheumatoid arthritis indication. You will start with the healthy population first. And then also definitely the PK characterization, any PK/PD characterization where you will establish a proof of action. Once we have this basic understanding, then the proof of concept studies are designed primarily with the range of dose, which should cover the dose which are close enough to efficacious, but at the same time, you have enough range so that you can characterize the exposure response relationship. So POC is very important in terms of exposure response characterization. But again, sometimes these POCs are conducted not from the pivotal clinical endpoint point of view. They are primarily conducted with some surrogate efficacy markers. So again, it all depends on how quickly you want to have this result so that you can start or pivot to the larger phase 3 trial. Phase 3 trial is actually, most of the registrational studies are typically phase 3 studies where you will evaluate pivotal clinical efficacy against the standard of care. Now, once we have a successful phase 3, and then that's eventually the registrational package, which I will highlight more on what it means from the clinical pharmacology to support such a registrational package, so that regulatory agencies can evaluate the safety or the benefit risk profile of agent to make that drug to be available for general population. After approval or the first indication, there are a whole host of the post-approval phase. So in industry setting, what you will see is not only you are completing a new drug registration, but you have to do the life cycle management of that program. And what it means is most of the time is either expanding the different indications or looking for some kind of post-marketing commitment that regulatory agency may give you. For example, let's say if some of the characterization during the drug development was not taken in detail, they may issue some post-marketing commitment. Or you can also do, most of the time, you will be like, for example, you will, let's say if your program is started with intravenous formulation, for the patient convenience, you may switch to the subcutaneous. So most of the new drug delivery system will be, will be conducted during the post-approval phase. Okay, so now switching gear to the what is clinical pharmacology? Clinical pharmacology is a translation science in which dose, exposure, response, and in this case, response covers both safety and efficacy relationships, are applied in context of patient care. And how do we do that? It's that's the real science. But idea is, or objective is to basically prescribe a right right drug, which is drug development objective, but more specifically for ClinForm is the right dose at the right time to the right patient. So, and I will go in each of these, what it means, like right dose, right time, right patient, what it means, and how, some of the clinical pharmacology characterization can cover these aspects in the final ClinForm package for registrational studies. Okay, so now this is a very classical picture. Reference are cited here, but when we have to relate the dose, exposure, and response, typically this is how it goes, right? So what you're seeing here is on the top left is a pharmacokinetic profile, which just an example pharmacokinetic profile. Based on the shape of the curve, you can guess that it's, for example, it's an oral dose, and you will collect the different samples, time courses, intense PK sample to characterize the full profile of drug early in the program. So once you have this PK concentration time profile captured, similarly, what you will do is you will try to also collect some biomarker, which I call it here as a pharmacodynamic. So what you will see is and on the right side, on the top right side, what you're seeing is x axis is a concentration, y axis is a effect. For example, let's say if giving you example, let's say if this particular drug is, let's say, cardiovascular, or let's say blood thinner, right? In the effect is the clotting time. You can measure the clotting time on the y-axis. So what you're seeing is, so in this case, as concentration increases, depends on the mechanism, whether the clotting time increases or decreases. That's what you will measure it. That's the PK. That's the PK-PD relationship. And once you have both of these captured, you will be essentially able to define the PD time profile, what you observe in the what you observe in your clinical trial. So any effect, and just to tell you, the PK profile is a function of dose, right? So let's say after given dose, you will have certain PK profile. Higher the dose depends on the dose linearity. You will see the proportional increase in exposure and so on. And then which in result will have a differential effect on pharmacodynamic. And again, once you have these relationship established, what it does is it helps you to really optimize your dosing regimen. For example, in this plot, what I'm showing is once you have a PK-PD relationship established. So for example, the plot on the left, what you're seeing is exposure response, and there is a sigmoidal relationship with efficacy. So the higher the exposure, it follows the, let's say, sigmoidal Emax relationship, and after certain exposure, there is no further gain in efficacy. But let's say if you see the red curve, it's hypothesizing it's a toxicity relationship. So as you are increasing the exposure, the toxicity is increasing exponentially. But ideally, what you will try to do when you're looking or optimizing your dose or making or prescribing a dose, what you want to do is you want to have define a therapeutic window and try to maintain the exposure within that therapeutic window, and that will be our dose recommendation. And that dose recommendation is primarily once you establish this exposure response relationship. So until unless you establish this relationship, making such a dose recommendation is hard. Most of the time, those dose recommendations are not optimal. So it's very important. On plot on the right side is actually giving you example that characterization of PK profile. For example, here the solid line is a population mean, which is, let's say, average concentration time course in the general population. But let's say that population can have many different variability. So for example, let's say if within that population you have a subject who has a hepatic impairment, and the drug in question is undergoes some kind of hepatic metabolism, what you can expect that subject to have a higher exposure, right? And so although your population mean is within your therapeutic area, therapeutic window, there could be some subject who may cross, let's say in this case, the maximum Cmax start showing some safety concern, then the subject who has hepatic impairment may breach that kind of safety limit and may show some safety signals. So, and that's what the ClinPharm PK characterization into different spatial populations such as hepatic impairment will try to look for these spatial population more closely and see if there is a larger difference in exposure which may be cause of concern. So now, so as you all so far in the slide, what I've shown is, I think there are key things that we definitely need to have a well characterized to really build up. That's the foundation of ClinPharm package. First is the pharmacokinetics, and I think most of the undergrad programs you have gone through this pharmacokinetics, like characterization, what it means. But in practice, how we do it is during the drug development is also, there are some nuances around it, right? So ADME profile is measured drug concentration over time. So first thing I think I did not touch upon so far is how do we quantify drug concentration. And again, I will highly recommend to follow some of the guidance, FDA guidance around the bioanalytical assay development, and how there are many different assays, and again it depends on the large molecules, small molecules. For example, when I was in CDRI, I used to have LCMS-MS for the small molecules. So there are different guidance. So it's very important to have accurate and precise concentration measured in the blood matrix of interest. And this has to be, most of the time, these characterization happen early in the program, like phase 1, mostly in the phase 1, and the phase 2, you will start limiting your PK sample collection in patient population. And in phase 3, generally what we collect is a very sparse PK. So for example, we will collect one sample per dosing interval, and one sample at the, let's say after 5 or 10, let's say at a steady state. So it's a very sparse PK sample because these trials are very large, and we don't want to make a patient have extra visit just to collect a PK sample. So most of the time, these PK characterization occur very early in the program. And it's important to have that so that we can design our phase 2 studies more appropriately. So PK characterization is not just mean like what is the PK profile in the given patient, but also most importantly is to understand what are the different factors that affect PK. So for example, basic demographics such as age, gender, body weight, race, and other factors such as any co-medication or any organ dysfunction such as hepatic or renal impairment will be assessed thoroughly. And there are very sophisticated tools to do these kind of modeling. I have listed the software like Vinolin, which is a non-compartmental that non-compartmental modeling does not do the covariate effect assessment. It's primarily the nonlinear mixed effect modeling, such as non-MEM or SimSip will try to understand the physiological basis for any effect of these demographic or let's say organ dysfunction on the PK. and more or so, I think one of the expectation for any clinical pharmacology is to have very good very good hands-on training on all of these tools, or at least few of these, because this is the foundation for any clinical pharmacology study. Next is the pharmacodynamic. Pharmacodynamic depends on the how you are in the industry setting. Typically, there is a dedicated functions called the biomarker or translation medicine group, who will be expert in basically working with the preclinical biology person to really understand the mechanism of action. But all of these, in context of registrational package, all of these learnings goes under the clinical pharmacology package. So as a clinical pharmacologist, one has to be have a very good understanding on the mechanism of actions, and what are the different pharmacodynamic measure one should collect so that some of this PKD relationship can be understood early in the program, to make, for example, the dose recommendation for phase 2 studies and so on. And again, the similar tools can be applied. Similar kind of covariate effect assessment for pharmacodynamics can be applied as similar to the pharmacokinetics. And once you have this pharmacodynamics collected, for example, let's say if blood pressure is a marker, right, for anything, I think having a blood pressure collected over the different time period will provide you the concentration. If the drug has an effect on, let's say, cardiac functions. Let's say I'm just giving you an example that blood pressure could be one of the pharmacodynamic marker, which can allow you to find any. Again, this is a pharmacodynamic marker can be for efficacy as well as for safety point of view. So you can have some kind of therapeutic window way of assessing your optimal dose regimen for your phase 2 studies. Now, I will go a little bit more detail in what the clinical pharmacology strategies are during the drug development. And again, all of these terms, there is a whole host of science behind all of these terms, and there are specific guidances, regulatory guidances around these terms, how to conduct these assessments. And again, some of these assessment may vary, depends on the small molecule and large molecule. So I think I mentioned this earlier on the preclinical, right? At the preclinical stage, at the time when the molecule at the preclinical, there has to be a lot of other activity that will support the clinical development. For example, you will have appropriate formulation. Again, the whole formulation development is very critical when you go to the clinical, because it can have an impact. For example, let's say if you start with a formulation which is not an optimal, and most probably most of the formulation what you have in early clinical, that's not the formulation you will have an approval, right? So most of the time we will have a clinical pharmacology package to support the bridging from early clinical formulation to the late stage clinical formulation. So we partner with whole pharmaceutical pharmaceutical formulation group to understand what are the changes has been made in formulation. Some of these changes can be small, which can be justified with the analytical comparability, but some of these changes could be very big. For example, if it's a tablet versus capsule, then you will be doing some bioequivalence study and so on. So the clinical pharmacology package very much depended on a lot of things what has been characterized preclinically. As I said, the bioanalytical method, again, it's very critical. And again, there are guidances. Sometime what can happen is these bioanalytical method can also change over time. So giving you an example, let's say you start with a bioanalytical method without knowing what could be a concentration in human, right? Because you have no idea when you're developing this method, what could be a concentration. Pretty much everything is based on the predicted PK profile from the animal model to the human. That's just a prediction. But in practice, what could happen is, let's say the drug undergoes hepatic first pass, very severe hepatic first pass, what you expected, then you can reach to a concentration level which is very lower than your lower limit of quantification in your bioanalytical method. So you may have to change your method and make it more, I would say, sensitive to measure some of these low concentrations. So again, so a lot of these things, what you are doing can change over time, and there has to be clean form package put together in a way to address some of these changes over time. IND tox safety, again, I think, as you all know that before we do in the clinical, any experiment, we have to have IND tox established. We have to have some of the novel concentration doses established, which will be a margin, one of the important factor to understand what should be the first in human dose. At the same time, also to understand if there is any safety liabilities that we should be monitoring in our clinical evaluation. For phase 1 studies, I think, as I mentioned, the idea would be to basically the idea would be to evaluate different doses and the schedules, and try to understand the PK characteristics. If it's a large molecule, we will also try to understand the immunogenicity, and I will go in little detail about this. We'll also try to understand, let's say if it's a small molecule which is given orally, you will try to understand the food effect. If most of these new molecular entity has to have some kind of QT assessment done, because cardiac safety is very important. If there was some preclinical finding associated with the cardiac, I think it become very important to have some dedicated thorough QT assessment. But for example, in large molecule, we don't do any thorough QT. We can just simply collect some of the QT during our phase 1 studies, and given because large molecule typically does not go through the cardiac toxicities or the cardiac I would say receptor interactions in a way. And based on these phase 1, as I said, PK/PD relationship, you will try to establish a phase 2 dose. During the phase 2 trial, definitely, I think having a proof of concept with more clinically meaningful clinical endpoint. There will be some other clean form studies such as DDI studies, hepatic renal impairment studies, as well as some of more intense modeling work such as population PK exposure response analysis will be conducted to inform the phase 3 dose selection. And phase 3 dose selection is the pivotal trial dose selection, and most of the time the doses what you study in phase 3, we will be looking for an approval for that dose. So the phase 3 dose selection is a big deal, and it should be it should be robust, as much as robust as possible based on the phase 1 and phase 2 observation. Let's move on. And what this slide does is it just shows similar things what we have seen early, but showing in some kind of interdependency. For example, let's say if you have in vitro in preclinical, let's say you have an in vitro metabolism transport characterization, that can inform you what kind of DDI study you need to conduct, right? Let's say if your drug is a CYP3A4 substrate, then you need to plan for DDI study, which can really establish the differences in PK in presence of CYP3A4 inhibitor or inducers. Similar thing, they will also for example, the mass balance study has to be conducted in the phase 1 to really understand what are the major route of elimination for a small molecule. And depends on those mass balance study, you may be conducting PK in a special population, such as, let's say, if you have a small molecule that is going through, let's say, 70% renal excretion, then the PK in the renal impairment patient become very important. And it could be vice versa for hepatic impairment as well. Okay, so what this slide is showing is pretty much the complete clinical pharmacology package for a small molecule, that by the time we are going to, let's say, regulatory agencies for an approval, this is what you need to have already characterized. Any question? Okay. So this is what you want to characterize while before you are going to regulatory agencies, right? So, for example, in a small molecule, you will typically have single ascending dose, multiple ascending dose PK characterization, which will give you an idea about your dose, whether the exposure increases with the increase in the dose, what is the accumulation, is your PK linear, and what is your steady state exposure, and so on. You will establish whether the PK is, let's say, if it's a non-oncology, you will try to establish what is, how the PK differ between healthy and patient population, mass balance study, and again, special populations such as renal impairment, hepatic, and sometime you will also require to have a pediatric development plan, even though you're looking for an approval in adult. Depending, most of the regulatory agencies require the pediatric investigation plan approved before you can do submission. So you will also have plan for pediatric population. Drug interaction, population PK, biomarker, spatial safety. I think these are all standard package that you will try to characterize before you go for registrational approval. Similar on the right side is the exposure response. Most of the time, the dose selection and optimization, when particularly looking for registrational study, you will try to do exposure response with your clinical endpoint rather than just limiting to the phase 1, phase 2 pharmacodynamics, so that when you're making a final dose recommendation, you can make a benefit risk assessment from the exposure response point of view. Again, depends on what is your formulation. You can have different bioavailability, bioequivalence studies in your clin pharm package. If it's a small molecule, you will have a food effect assessment. Again, depends on molecular characteristic of your agent. Let's say lipophilic drugs tends to have much of the food effects. So it's good to understand the PK concentrations changes when it's given in a fasting condition versus food condition, so that you can make an appropriate prescription decision for drug. And again, standard in vitro characteristics such as protein binding, in vitro drug metabolism, transport, and the drug interaction studies are part of a standard clin pharm package. Bioanalytical assays, typically there is a dedicated module where all of these bioanalytical assessment will be reported. So these are very critical for any successful clin pharm package. Now, I think I can skip these. Most of you may be familiar with the small molecule, large molecule differences, but just give you the reason why we have a differences in package is primarily because of the difference in the PK characteristic with these agents. For example, you can have a small molecule. A small molecule tend to have a lower molecular weight than the monoclonals. When after let's say, for example, oral administration or IV administration, in case most of the monoclonals are given subcutaneously or IV, They demonstrate a low volume of distribution, primarily because of the molecular size. Large molecules tends to limit into the central volume, which is your peripheral blood. Half-life, again, since small molecule has tendency to go to the first pass or renal elimination, they tends to exhibit shorter half-life, while the large molecule will have longer half-life because it does not go through any renal or metabolic elimination. And again, half-life has an impact on what should be the dosing frequencies. For example, most of the small molecule typically will have once a daily or maximum a week or so, but it depends on what is the PK/PD profile. But mostly the large molecules typically have a long half-life, and most of the time they are given either every 2 weeks or every month. Those kind of dose regimens are typically prescribed for the monoclonal antibodies. Clearance, again, the similar concept there. Renal elimination, again, I just touched upon that. Given the size, large molecule may not able to filter through, kidney. And metabolism, again, the large molecule does go through the proteolytic degradation, which are typically is not hepatic or renal. Other thing for the large molecule, which is not relevant for a small molecule, is the immunogenicity. And again, I think the concept of immunogenicity is very similar to what you have in a vaccine, but here it's a little bit more negative sense. So, for example, any biological entity, when administered in the body has tendency to form antibody against that foreign moiety, and that's what is basically immunogenicity. So, any anti-drug antibody formed when any biologic is administered is basically, we try to quantify it, and that's what we call it immunogenicity. Why it is important? Well, some of these immunogenicity can cause infusion reactions. So, excuse me. So, for example, let's say many drugs early on, when monoclonal antibodies was start coming in drug development, these were chimeric, means there was a significant portion was non-human source. So what may it lead to? The most of the time body will activate the immune system against those foreign bodies. So most of these antibodies tend to have a higher immunogenicity early. Nowadays, most of these monoclonal antibodies are fully humanized. In a way, there is a very or pretty much no chimeric portion in monoclonal antibody, and nowadays what you see, the immunogenicity with large molecules tends to have like limited, but they do occur. And sometimes these antibodies, anti-drug antibodies, can be neutralizing. What it means is they can bind to the monoclonal antibody at the site which is important for your biological activity. And that may result into the lack of efficacy. Basically, your monoclonal antibody become inactive once your anti-drug antibody bind to the monoclonal antibody. So, and actually that's a concern for efficacy. Means if you have anti-drug antibody which is neutralizing, means it is binding to a site which is responsible for activity. This will lead to a lower efficacy, and that could be a concern. So it has a both safety implication as well as implication on efficacy. So similar to PK characterization, immunogenicity characterization is also very important for biologics. Now, clinical pharmacology package for large molecule. I think the earlier I showed one with a small molecule, and what I'm doing here is I'm crossing out some of the other few studies that you will not be conducting with the large molecule because those are not relevant. For example, you will not require to do a mass balance study, as I said. It's primarily monoclonal antibody does not go through renal or hepatic elimination. Additional thing what you will do is immunogenicity, just which I just spoke. Again, hepatic impairment, renal impairment, there will be limited assessment. You will not be conducting a dedicated study, so to speak, but you will try to understand if there is a difference in PK. And the reason being that sometimes these renal impairment, for example, there are certain disease conditions where your renal filtration system can be completely or less severely damaged, which can increase the pore size of your glomerular filtration. For example, lupus nephritis. We have very good experience with that. One of the monoclonal antibody we were testing actually was able to filter through the kidney in the lupus nephritis patient, and we were surprised to see that because it was monoclonal antibody, and and the drug was improving the lupus nephritis prognosis in a way, and the clearance was basically decreasing over time. So as the disease was improving, clearance was decreasing. So there are exceptions where you can have some disease condition which can lead to renal filtration of even small monoclonal antibodies. Drug interaction, again, there is for large molecule that does not go through CYP metabolism, but there are new literatures recently where it's reported that large molecule which has potential to do a cytokine modulation may lead to indirect CYP3 induction or inhibition. So there are some indirect drug interaction potential with the large molecules. So I think that typically the way to address is to understand what is the cytokine modulation potential with the large molecule, and depends on the cytokine modulation, one can argue in against any drug interaction potential. So there are ways you can still assess those cytokine mediated DDI. Again, as I said, with the large molecule, there is no requirement to do a thorough QT, and typically some assessment of QT during the phase 1 study is sufficient to establish any cardiac QT related liability for large molecule. I think for bioanalytical method, as I said, aside from the PK, there is another critical component, which is anti-drug antibody and the neutralizing antibody assays to characterize immunogenicity. Okay. So this is one of the examples that, again, immunogenicity, there are how to assess the effect of immunogenicity on PK and let's say safety and efficacy. We have publication in AAPS General. Please take a look on. We presented how should we assess the effect of immunogenicity on different let's say PK, safety and efficacy. And one of this plot, so this is basically a real dummy clinical data, so to speak, but it's very close to what we observe in clinic as well. So what you're seeing here on the x-axis is a trough concentration. A trough concentration means the minimal concentration after, let's say, any dosing interval. Let's say if it's a Q2 week, it's a trough concentration at, let's say, day 14. So that's what you're seeing in the. So this is a trough concentration. at, let's say in this case, the drug was given, let's say every 8 week, or it was a weekly. So this is number of weeks, right? So let's say it's the trough concentration at every 8 week or so. So, and each of these concentration may represent the different patient trough concentration. So what you're seeing is the red dot with the different size of red dots are basically the anti-drug antibodies. And the size of this circle represent the basically the concentration of your anti-drug antibody, which is measured as a titer. So higher the anti-drug antibody, the bigger the circle. So what essentially what you're noticing is most of these red circles are basically the way we plotted is we always collect the immunogenicity and the PK together. So let's say if subject come for visit and we have to collect immunogenicity, we also collect the PK, corresponding PK. So always the PK and IMG is always matching. So what you're seeing is for all of these observation, which is a blue dot, the PK observation, you have overlapping immunogenicity observation. So in subjects where you have immunogenicity observed, what you are noticing is most of these subjects where you have immunogenicity, the concentration was low, and most of these subjects, the concentration was below LLOQ. So this plot exactly reflect that when you have higher immunogenicity, PK is getting affected by immunogenicity. And what is the mechanism of this, right? So there are many mechanisms. One of the hypotheses is that when drug and antibody makes a complex that is more and bigger size, it's more prone for the phagocytosis, or let's say, for example, proteolytic degradation. So eventually your concentration will decrease. And this is what we are seeing in this case. For example, here, most of these ADA positive, the time point where we have ADA positive or anti-drug antibody immunogenicity was positive, the concentration was very low, and which could be a result of immunogenicity. Okay, so now I will skip this slide and okay, so I think what I'm doing here in now next few slide is I will give a little bit more detail on the what are the clinical pharmacology studies are, how they design, what is the objective, and so on. So single ascending, multiple ascending dose for ClinPharm, I think for us is really the PK characterization. We try to understand the variability. We try to understand what is the dose linearity or dose proportionality. We try to understand the steady state PK parameters such as accumulation, any time dependency, and also try to understand what are the, let's say, drug elimination pathways are. This is typically done in major non-oncology setting, is typically done with the healthy volunteers. And most of the time, the doses are determined using the preclinical toxicology studies. The next is the ADME mass balance study. The objective of this study is primarily to understand the mechanism of clearance and portion of parent drug conversion to metabolites. Again, it's typically done with the single dose in healthy male, 4 to 6 patient healthy males. With the intended route of administration, it will be done with the, let's say in this case, radio-labeled drug molecule to determine the amount of radioactivity in different matrices such as plasma, urine, feces. Hepatic impairment, I think the objective here is to understand the effect of renal impairment on the drug clearance, and to make a dose recommendation for various stages of renal impairment. So typically you will try to enroll a patient stratify with the different renal stratification with the normal, mild, moderate, and severe. Again, there are dedicated FDA guidances or regulatory agency guidances that one has to follow for the best practices. Similar for the hepatic impairment, you will try to stratify with the Child-Pugh class A, B, and C, so that you can make a dose recommendation in this population. Okay, so for drug interaction, and most of the majority of ClinPharm package is actually these drug interaction. If there is a big liability with a small molecule, you will have a many different combination to really assess what is the effect of different concomitant medications on the PK, or if depends on whether your drug is substrates or inducer or inhibitor. But typically it's done with, again, healthy volunteer. Crossover design is preferred, but if it's long half-life, the parallel design is conducted. Thorough QT is typically done with the supratherapeutic dose to establish to identify drug that prolong the QT. And again, there is a guidance on this to typically, if your QT prolongation is defined by the 95% CI upper bound greater than 10 millisecond, this would require, if you have this kind of QT prolongation, it will require thorough ECG monitoring in the pivotal trials. So it has an impact. So I think this is typically done early in the clinical development so that you understand the QT liability. Okay, I will skip this. And I will present one case study, cabozantinib. Again, this is, this will show how the clinical pharmacology package finally result into some of the dosing recommendation. So cabozantinib is a kinase inhibitor. Again, it's a small molecule. It's been approved for renal cancer and hepatic cell carcinoma. The approved dose is a 60 milligram daily oral dose, and it is recommended 1 hour, either 1 hour before or 2 hours after meal. So in their single ascending and multiple ascending dose, they have established that there is a 4 to 5-fold mean accumulation. So means after repeated dosing of, let's say, 60 milligram, you will see the 4 to 5-fold higher exposure. And at a steady state, and typically the steady state was achieved at day 15. So means there is no further accumulation after that point. And in mass balance study, since it's a small molecule, they have conducted mass balance, and the 81% radioactivity was recovered, and 54% was in feces, so which highlights that it's mainly the hepatic elimination. 27% was in urine, so there is a lesser portion into renal elimination. And as expected, the renal impairment study concluded that there is no dose adjustment recommended in mild or moderate. Typically, severe renal impairment patients are very limited, and most of these trials, when we conduct, there is a limited severe renal impairment patients to make a conclusion. Hepatic impairment, as expected, they have seen the exposure was increased by 63% in patients with the moderate hepatic function. Hence, the dose recommendation was to reduce the dose from 60 to 40 in moderate hepatic. So you can see that some of these ClinPharm studies actually directly inform the final dose recommendation, what you see in the label prescription. Now, as I said, the drug interaction, again, for this particular drug, this was a CYP substrate. So they have to conduct the studies with a strong CYP3A4 inhibitor as well as strong CYP inducers. And as expected, the ketoconazole, which is a strong CYP3A4 inhibitor, resulted in increase in cabozantinib exposure by 38%. So for this particular category, so let's say the recommendation, if you cannot avoid a strong CYP3A4 inhibitor, you would recommend to reduce the cabozantinib dose to 40 milligram, because this inhibitor may result into higher exposure of cabozantinib. And vice versa, if you cannot avoid strong CYP3 inducers, you need to increase the cabozantinib dose to 80 milligram. And most of these findings are based on the clinical pharmacology studies, as I mentioned earlier on. And again, there were some other CYP enzyme, PP enzyme, which was also found in the preclinical studies. So they have conducted some clinical study with the CYP2C8 as well as PPI in their clinical trials. So again, non-clinical package informed what should be conducted in the clinical pharmacology package. Okay, with the food effect, since you see there was a recommendation to take it 1 hour before or 2 hours after, it was primarily based on their food effect study. Again, there is a whole guidance how to conduct a food effect, what should be a specific meal and so on. But as you can see, the AUC, which is the area under curve of cabozantinib, was increased by 41% with the high fat meal. So it was recommended not to administer. So they have given window 1 hour before or 2 hours after. So the next slide here, what I'm presenting is again one of our internal example, which was published in Annals Oncology, very high impact journal. This is an example of post-approval application of ClinPharm package, right? So once you have characterized your clinical pharmacology, you can use those characterization to extend or make a dose recommendation without any clinical data. So this is an example where one of oncology drug, which was initially given, let's say mg per kg basis, which means the patient, let's say if you have a 70 kg patients coming, the physician need to weigh the patient, determine the dose, and then administer the dose. But what we have done here is, based on some of these PK exposure response characterization, we have shown that it really doesn't matter if subjects who is, let's say 60 kg or subjects who is 100 kg, 110 kg, it really doesn't matter if you give them flat dose. So overall, I think what we have done is we have made the dose recommendation switch from mg per kg to the flat dose based on the PK modeling. So what you're seeing here in this plot is panel on left side is the 3 mg per kg, which was the initial approved dose, and on the right is the flat dose, which is the new proposed dose purely based on the modeling data without any clinical. So what you're showing here, the exposure, predicted exposure in x-axis is the different body weight patients. So let's say patient from 40 kg to 160. What you're showing is if you give a mg per kg dose, exposure increases with the increase in body weight, right? But if you give a flat dose, exposure decreases with increase in body weight, because let's say heavier body weight patient, if you still give them 240, you will have low exposure. But what this plot is showing is that mainly from the safety point of view, the increase in exposure in low body weights. For example, let's say if you see the flat dose section, the lower body weight 40 kg subjects exposure is still below the highest dose what we have tested in the clinic. So essentially what we are saying is, if we give flat dose, there is no safety concern in low body weight patient, right? But one can argue what happened with the heavier body weight where you have a low exposure. So this subject may have lower efficacy. And that's where the PK/PD or exposure response relationship comes into picture. So with this particular drug, what we have established is, for these exposure range, there is a flat exposure response relationship. What it means is, the subject who has exposure at this range versus this range, there is no differences in efficacy. So having the exposure response relationship and PK characterization, essentially we made a dose recommendation without any clinical data, and this flat dose is already approved across different countries around the world. So this is one example where the proper characterization of clinical pharmacology can help in the post-approval setting. And I will pause here. I think I am up 11:30 already. Vivek, I will pause here. Maybe I can take a few questions if there is a time.
Okay, sir. So thank you very much, sir. And it's the time for question hour session. So if some attendee is having question, you can WhatsApp me or you can type in chat box also. So sir, one question I have received is that what are first in human studies? One of our attendee student has asked that what is first in human studies, and can you elaborate that how these doses are decided from animals to humans like that?
Mm-hmm. Yeah. So first in human study is basically, as I mentioned early on, first in human studies depends on which therapeutic area you are working on. For example, if you're working in a non-oncology setting, the first in human study will be typically done in the healthy volunteer, healthy human, healthy volunteers. This study is designed mainly to assess the single dose tolerability, means the safety. And again, you will have a range of dose, basically 7 to 8 different dose cohorts, which will be designed in a basically either 3+3 setting, means you have to have a safety established with the lower dose before you go to next dose. So it will be a tiered dosing approach in a way. So that's the first in human. And how do we select the dose is again depends on the small molecule and large molecule. So for example, so first in human dose selections are, for example, if it's a small molecule and you have PK characterized in appropriate preclinical, typically it's preclinical, they will conduct at least 3 different animal species, rat, dog or monkeys, and then there will be allometric scaling. There are different approaches where you can make a dose recommendation. So there will be allometric scaling. At the same time, so that will help determine on, okay, what is the equivalent human dose. So that's one concept. What is the equivalent human dose, right? The second concept is what is the human efficacious dose. That is typically based on your PK/PD characterization in the preclinical aspect or the in vitro binding affinity. And then the other aspect is what is your minimum or let's say the safe starting dose, and that will account for the safety margin what you have established in your IND tox. So there are 3-4 different approaches, and what you do is you take the most conservative one. So you will have a dose recommendation from all these 3 different point of view, and then you will pick the most conservative one. For large molecule, you will also do minimum effective dose, which is a Mabel approach. And again, there is a whole range of science behind this. I would highly recommend to follow, some of these FDA guidance and some of the recent publication that came out through the IQ consortium on how to select the doses for large molecule, what is the Mabel approach, and so on.
Any other questions?
Yes, sir. Sir, I have one question.
Yeah.
Yes, sir. Sir, thank you. First of all, you have covered all the aspects of clinical pharmacology very nicely. Sir, Actually, we are formulating one topical formulation, and where we have used our target molecule is a antimicrobial peptide. So challenges which we are facing before a challenge like BRC committee and all that, they say that peptide molecular weight, and second is its immunogenicity. So they say that peptide is, sir, our peptide is 6 kD. So, sir, how to overcome this? Because it has got very good antifungal properties, and it is effective against resistant fungal strain also. So they say that molecular weight is very high. So how to overcome this, and what are the challenges which are associated with giving this kind of biologics, peptide and protein molecules, like their permeation study or diffusion studies? So how to justify this, sir?
Yeah. So first of all, immunogenicity, right? Again, in this case, the immunogenicity for most probably what your experience is basically skin reactions, hypersensitivity. It's not a systemic immunogenicity, so it's a little bit different concept.
Okay.
So so they can call it immunogenicity in terms of your skin rashes, hypersensitivity, those kind of things. So it's inherent. I think there are some system model which can, based on the peptide sequence, they can determine how likely they will cause immunogenicity.
Okay,
there is
sir.
there is a whole consortium on the immunogenicity characterization. So I would highly recommend, based on your sequence of peptide, they can establish this is the wrong sequence. Maybe you need to tweak this little bit. So that's one thing. So construct of peptide itself can be assessed. In terms of the KD or the molecular size,
Yes.
I think the permeation is important, right? So I don't know, in your formulation, there are ways you can do permeation enhancer in a way which can allow that. Other than that, I don't think you want to have any let's say slow release implant, or which can allow to have more time. Let's say, if there is some saturation, right? There could be something like, if you can put an implant which can have more prolonged period of interaction with your skin, which will allow slow but sustained absorption. These are the two approaches I can think of. But I think most important is the safety, right? If you have your peptide construct itself is causing irritation, and I am suspecting it's not because of formulation, it's not because of some of the other formulation component, and which is again very important to check for these topical formulation. If some of these co-formulating agents are off limit, can also cause skin irritation, and maybe those could be a reason, not the peptide itself. So
Okay, sir.
Yeah.
Sir, and actually, if it is a naturally occurring peptide, then, for example, if it is a salivary peptide or it is a human neutrophil peptide, if our targets are those, like it is basically it is an antimicrobial peptide and is having natural origin. They are found in our body, and they are reported to have a very good antimicrobial activities.
Mm-hmm.
And
Yes,
sir.
so if it's a human, I think then the immunogenicity should not be the case. I think most likely it could be due to the formulation itself.
Okay, sir. And sir, suppose it is more than 500, like for transdermal drug delivery system, they say that molecular weight should be 500, not more than 500. So peptides, obviously it will be more than that. So we are not targeting to systemic circulation, but to the topical local. So is that justification is correct? If we formulate it in a formulation transdermal, and as you said that we should have a sustained release like in implants, or we can have vesicular drug delivery system. So can we justify it in this way?
I have to see. I have not exact experience with the topical formulations. I have to see, was there any case experience? Is there any I'm sure I hear most of these nowadays, like you will see even in the cosmetic, right? They marketed with the serums, right?
Yes,
I am
sir.
expecting these are peptides in a way.
Yes, sir.
So, but definitely they may have gone through kind of, some kind of limits which have allowed them to have a effect and at the same time permeation given
Yes.
that. So I would definitely start from there. If there is any already a marketed product with such molecular size, and what was the strategies in place, right?
Yes, sir.
So typically these reviews are publicly available
Okay,
for
sir.
any of these marketed products. So I will start from any marketed product example, and
Okay,
then
sir.
try to look into what strategies they have developed. Again, it all comes down to strategies, right? What kind of studies we need to assess and so on. So having any example of marketed product and try to look into their ClinFarm package or their biopharmaceutical characterization package, that will give you a very good idea about how to navigate with this issue. Okay, sir.
Yes, sir. Thank you, sir. Thank you very much.
So there are no other questions, yes, sir. You have covered everything very nicely. And
Thank
sir,
you
may I
very much.
request you to close your presentation mode?
Yeah. All right.
Yes. So on behalf of Parul University, I thank Dr. Satyendra Suryavanshi. And sir, it was very interesting and a great learning to hear from you. It was really very informative and comprehensive, and you have given a very deep insight into the drug development process and the key clinical pharmacological considerations. And also you have given a deep insight into the small and large molecule drug development. And sir, I can understand that the time over there in New Jersey is 11 past 30 night. So that tells about your eagerness to speak at the event and your enthusiasm to speak in this webinar. So sir, thank you very much for sparing time and joining this webinar as a speaker. We are really thankful to you for your talk on this very important topic, sir. So, on behalf of the Parul Universities and Parul University family, sir, kindly accept this certificate as a token of gratitude from us, sir. Please accept this certificate.
Thank you very much, Vivek and Dr. Jha. It's very kind of you to issue such certificate. I'm looking forward to have more interaction, students particularly, joining and expanding their clinic, expertise in clinical pharmacology area.
In fact, it was a time frame for us right now, sir. But in future, we would like to have you again for because I can understand some important topics are still we should learn from you, sir. So in future, also we would like to have you on the webinar.
Yeah,
Parul.
sure, anytime. Yeah.
And thank you very much for sparing.
Thank you. Thank you. Have a good day.
Good day,
Bye-bye.
sir.
This is the end of our webinar, and thank you all of you for joining us today. Thank you, sir.
See you. Bye.
And your feedback is very valuable to us as it gives us a guidance how to make our future webinars better and better. So I'm posting a feedback link in the chat box. You are all requested to fill the feedback form, and certificates will be provided to all those who have given us the feedback. So here is a link to the feedback form.